Dear students, in this module, I'm going to talk about the basic underlying algorithms that are used to classify the protein structures. You already know that various databases and schemes exist, such as the CATH, the SCOP, the FSSP, as well as the P-class. So these schemes help you classify the structures of proteins and look at the families, the superfamilies, the folds, and classify them in terms of their function, their similarity, and evolution. So how exactly do we compare the two protein structures or a structure within a single protein? Towards that, several algorithms exist, and we're going to see the philosophy of these algorithms, and there are actually just two basic methods. The first one is to see the intramolecular distances, and the second one is to look at the intermolecular distances. So let's look at both of them. So the first one is the intramolecular distance algorithms. So in this, the proteins are considered as rigid bodies in 3D using a Cartesian coordinate system, and then you measure the distances within the protein structure, and that is, in the case of the classes, the domains, there will be lots of hydrogen bonds within a specific domain, while there will be no hydrogen bonds with the rest of the structure, and therefore the distance within a domain, the intramolecular distance, will be small as compared to the distance between this domain and the rest of the protein. Examples of these schemes include the VAST and LOCK schemes. Next is the intermolecular distance algorithm. These schemes actually use the distance between different proteins. So in that, you have 2D projections of these proteins, and you're looking at the various torsion angles between the different atoms. So the structural alignment using internal distances and angles is performed in this type of a scheme. As I just mentioned, the basic idea of such schemes is to capture the internal geometry of the protein and then talk about the overall structure. The examples include the DALI and the SSAP algorithms, and we'll look at them later. So in conclusion, the intramolecular distances or the intramolecular distances are very useful to compute the overall structural similarity between different proteins or domains. So these algorithms, they compare not just the whole or the overall structure of proteins, but also help you to estimate their evolutionary distance. That is, how much these two structures may be different from each other and therefore close or far away in terms of evolution. Such studies also help you to understand the function of the proteins, because if the structure is similar, then most probably the function of these proteins is also going to be similar, and therefore such schemes are very useful.